epithelial layer so they can reach to the site where infectious agents are present, right? So this is about the transport. Okay, so movement of antibody across epithelial layer. And the antibody that are predominantly required to be transported at mucosal sites in respiratory system, gastrointestinal tract, and urogenital tract are the IgA type of antibody. Not to forget, in breast milk also you have IgA. Okay. And once they are there, so their function is to bind to microbes present in, for example, the digestive tract, right? And they can sequester, or they can actually bind to this microbe and not allow them to infect other cells, right? IgG antibody can also be transported from mother to fetus. Okay, there are different mechanisms for doing that. Okay, so the next thing is, so we'll talk about the transport in one more slide, exactly how this is carried out. Okay, now the classes and biological activities of different types of antibodies. How many different types of antibody do you have?
So five.
Five, yes. And so while the general function of these antibodies is to bind to the antigen, which might be present on surface of these microbes, or maybe a soluble product, maybe toxins, and neutralize them, but there are also some specific functions associated with the class of antibodies that you have. Okay. These are all those five types of antibodies, five major classes. We call them as classes of antibodies, or in other words, isotype, right? You have IgG, which actually is the predominant antibody isotype that you find in blood, right? The structure we already have talked about. So two light chain, two heavy chains. You have the hinge region, right? And the heavy chain that you have also has these different domains, similar to that you have in light chain. We already have talked about all those things, right? Now, why we call it as IgG is because of the heavy chain. And the heavy chain in IgG is gamma. In IgD, this is delta. In IgE, this is epsilon. IgA, this is alpha. And IgM, this is mu. So that decides these different five classes of antibodies, right? So IgG is present in circulation predominantly and in large abundance out of these all four isotypes. IgD is usually present on surface of the B cells that you have. Okay. Very rarely you find them secreted either in circulation or in tissue fluids. Okay. And that is why, so this type of antibody has not been characterized that extensively. So whatever the data has accumulated, that is based on whatever the minuscule amount you can get from either circulation with extensive purification protocols, and subsequently with the cloning and expression strategy, right? Because you don't have really too much amount of this antibody to be isolated from patients, right? Other class of antibodies is IgE, which is actually critically involved in these allergic reactions, right? One thing that is clear is that these antibodies, they have hinge region along with IgA, so it also has the hinge region. So three isotypes or three classes have hinge region, two classes do not have hinge region. IgE and IgM do not have this hinge region. So their flexibility of to move around actually is pretty limited, right? Uh, and that hinge region part actually is replaced by another domain. So essentially, so the domain in heavy chain that you have for IgE and IgM is one additional domain. Okay, so here you have up to 3. Okay, in the constant portion. Okay, so in variable portion you have whatever you have, right? So for example, here you have 1, 2, 3. Here also 1, 2, 3. So here you have 1, 2, 3, 4, right? And in IgM also you have 4 domains in the constant region of heavy chain. Okay, so as far as function is concerned, in allergic responses, IgA usually exists in dimer, okay, and IgM exists as pentameric version of this antibody. Okay, and how these things are brought about is for IgA dimer. So you have something that we call as J chain. J means joining. Okay, so there is one polypeptide chain which actually join these two antibody molecules so that it exists as dimer. So you also have this J chain in IgM, but the pentameric nature is brought about or caused by this disulfide bond between the C mu3 and C mu4 domains. Okay. Now you can see that in IgG, so you have only 2 antigen binding sites. In D also, in IgE also you have 2 antigen binding sites, right? So that means 2 paratopes. In IgA, when it exists in dimeric form, so you will have 4 of those, 1, 2, 3, 4. But in IgM, so you have 10, because you have 5 molecules and each molecule has 2, so that means 10. It can bind to 10 different antigens. The other thing is, this is one of the first antibody isotypes that you produce following immunization or infection, right? So the first antibody isotype you would detect following infection or immunization is IgM type of antibody. IgM along with IgD form this receptor on surface of these cells. Okay, IgD is rarely released. IgM is released from the surface, and then you can also find it in circulation, right? This is basically a comparative analysis of all these different types of antibodies that you have. Okay, more characterization. I understand this is a lot of information, and you actually there is no better way of learning it but to actually cram it up. Okay, so you just have to remember. These are different classes. How do you divide them in different classes? Based on the the heavy chain that you have in IgG, gamma, M, mu, A, alpha, E, epsilon, D, delta. And then these antibody classes also exist in different subclasses. Okay. For example, for IgG, so you have 4 different isotypes, 4 different subclasses rather. For IgG isotype, you have 4 different classes, right? Gamma 1, gamma 2, gamma 3, gamma 4. And this is based on the characteristics again of the heavy chain. And now we know that sequence also vary the sequence utilization. Okay, the amino acid composition in the heavy chain also decide these subclasses. If you think about it, how these different classes were discovered at that time, when the sequencing and molecular biological methods were not known. So this is based on the cross reactivity. So that means, because antibody molecule actually are protein molecules, right? So they can actually form epitopes which can be recognized by your B cells, not your own B cells predominantly. So for example, if you immunize IgG type of antibody and inject in, for example, rabbit or mice, and then you collect serum and try to see the level of cross reactions with different IgG purified fractions, right? So then you get these subtle differences, and those subtle differences are caused by these different subclasses. Some antibodies are going to recognize IgG with gamma 1 chain, some other with gamma 2, some other with gamma 3, and some other with gamma 4. And it also became much more easier when monoclonal antibodies were discovered. Okay, so we'll talk about what monoclonal antibodies are. Okay. So essentially, monoclonal antibodies recognize only single epitopes. Okay, single epitope, not the antigen. We are not talking about antigen here. It's about the epitope. If an antibody can bind to a specific epitope, okay, and you can actually uh, immortalize those B cells to produce that antibody. So this a B cell clone is going to give rise to monoclonal antibodies. There is a name to that technology. We call it as hybridoma technology. So we'll discuss what this hybridoma technology is all about. Okay. IgM, so you don't have different subclasses which have been identified. For IgA, so you have alpha 1 and alpha 2. For IgE and D, so none known, right? For light chain, for example, if you have IgG or IgM, all these classes, so they actually utilize either kappa or lambda as their light chain. Now, kappa and lambda means so actually there are different genes which encode for either the kappa chain or the lambda chain, right? So depending, and this actually is a very random event. So which chain will be present in which isotype? And this can also be touched upon when we talk about the antibody diversity, right? So kappa and lambda. So it's same everywhere. So depending on what light chain you utilize, um, and that also will have some characteristics such as what antigen this antibody is going to recognize and so on and so forth. More or less, the characteristics, the analysis on light chain is not that well known as is for the heavy chain. Okay. And the simple reason is very lately, actually, the approaches were discovered to isolate these light chains separately. And then you can characterize to what extent do they contribute to the antigen binding and to what extent they don't. And it was discovered, although the paratope is formed by light chain, heavy chain, 3 CDRs in basically light chain and 3 CDRs in heavy chain, right? But the major contact point to an antigen actually is contributed by the heavy chain. Light chain actually more or less provide as a scaffold or basically a framework. This is not to say that it does not have this variable region, but predominantly the interaction with antigens are affected by the heavy chain that you have, the variable region of the heavy chain, right? Now, depending on your gamma chain and kappa chain, so you can come up with a different formula, a formula to represent these different antibodies, right? For example, you can for IgG you can have gamma 2 because there are 2 heavy chains, right, and 2 kappa chain, and same 2 heavy chain and 2 lambda chain. Either. So this is always for one antibody molecule which is produced by a particular clone of B cell. This will be either kappa or lambda. So it cannot be both, right? For IgM, so basically again, you know this is the dimeric nature, and depending on whether it exists in monomeric version versus a pentameric version, so you can actually put this whole thing into bracket, and this is n times, and n can be 1 or 5. 1 means now actually this antibody is existing as a monomeric version, and 5 means this is existing as a pentameric version, right? For IgA, again, alpha 2, kappa 2, alpha 2, lambda 2. And although, so I said that this is a dimeric, this usually exists as a dimeric, but some reports have suggested that they can also exist in trimeric or even the tetrameric version, okay? But predominantly you find either as monomeric or dimeric. For IgM, you either find as monomeric and more predominantly as pentameric. And same is the case with IgE. Epsilon 2 chains of epsilon and 2 chain of kappa, or 2 chain of epsilon and 2 chain of lambda. And same is the case with delta. Is that clear?
Yes, sir.
Yeah. Okay, so something more about IgG. Okay, you have basically these are different depictions that you get for IgG, IgG1, IgG2, IgG3, IgG4. And you can see that hinge region actually is quite extended with a lot of disulfide bond between these two heavy chains in IgG3. Okay. And you can actually now see, because the length of this region actually is quite different in different classes. So major epitopes which distinguish whether this is IG1, 2, 3, 4 actually are present in this region, right? Most abundant antibody that you find in blood, or in other term in serum, is IgG. Almost 80% of the immunoglobulin that you find consist of IgG. Okay. Clearly defined based on the size of the hinge region, number and position of these interchain disulfide bonds, and this is the relative concentration Ig. So even in one individual, actually you can find all these isotypes, but if you calculate the relative concentrations in serum, so IgG1 is the most predominant, then you have IgG2, 3, 4, right? Although the heavy chain that you have is almost 90 to 95% homologous at DNA sequence level, okay? But eventually what amino acids are there, so that actually will give rise to these different epitopes which may not be present here but are present here. And that actually approach was used for dividing them into different subclasses, right? As far as function, so they can be transferred through a placenta and come basically in fetus and provide some level of protection in fetus, right? Or in a newly born individual, okay, neonates. So IgG is the antibody which actually provide protection to neonates, okay, because it can actually transfer through placenta. It can activate complement system, okay. So these are two things that you have to actually be more critical about, that yes, they can activate complement and they can be transferred through placenta. And most predominant antibody is IgG.
Excuse me, sir.
Yeah.
Oh, sir, in the previous slide, could you please repeat how they were differentiated in various types?
Depending on the amino acid composition that you have, and what amino acid will you have will give rise to different epitopes, right? So we'll talk about the epitopes present in an antibody, okay? Although you know that antibody molecule has a paratope which can bind to antigen, okay? But remember, at the end of the day, the antibody molecule is also a protein. And if it is injected in a different species, or in some cases even in the same species, so you can generate antibody against antibody, anti-antibody, okay? And those things are predominantly induced against the concentration. Okay, there are three types of epitopes and or antigenic determinant that you find in antibody molecule. We'll talk about that in separate slide. Yes.
Right, sir.
Yeah. Okay. IgM exists in pentameric version, monomer on the membrane, but when it is basically secreted out, so this exists as a pentamer. So this is the second most predominant antibody present in serum. First one is IgG, and then you have the IgM. Okay, 5 to 10% of total immunoglobulin, almost 90% is IgG. 5 to 10% is IgG or IgM, right? And this is the first antibody response in neonates and first antibody response after any primary response following an immunization or infection. Okay, it's very efficient in causing what we call as agglutination. Okay, so what is agglutination is basically when, for example, this antibody interacts with a particulate matter. Particulate matter can be a bacterium, a parasite. So basically this antibody can bind to different epitopes, and because of this cross linkage, so you actually can make what we call as agglutinates. Okay, how you basically distinguish agglutinate with precipitates is in precipitates, so you have more or less 1:1 stoichiometric interaction. Okay, for example, IgG kind of antibody, so it brings about precipitation. But IgM kind of antibody is more commonly able to cause agglutination. Okay, maybe in one of these slides that I'll talk about on the techniques, I can try to distinguish between the agglutination and precipitation reaction. Okay, but for the time being, just remember, when antibody interacts with an antigen in soluble form, this gives rise to precipitates. And when it binds to antigens, and if this antigen actually is in particulate form, so it gives rise to agglutination. This is very efficient in complement fixation. Okay, complement fixation means that actually it can bind the complement binding site actually is present here, and it can very efficiently activate complement system. It has this J chain, which is required for this joining. Okay, required for polymerization of monomer to form this pentameric and eventually bring about this disulfide bond formation, right? You also find IgGM antibody at mucosal site, but the most common antibody you find at mucosal site is IgA, right? So these are some of the characteristics associated with IgM type of antibody. IgA exists in dimer. Okay, this also depending on what parasite or what infection you have encountered. So this also can, this is more or less exists in the same concentration as you find for IgM. Okay, 10 to 15% of total serum IgG can sometime be constituted by IgA. But this antibody is predominantly present at mucosal sites. Okay, we already have talked about it. This can exist in monomeric, dimeric, trimeric, or tetramer. Most common form that you will find for IgA is dimeric, right? So this antibody you will find in external secretions, for example, breast milk, saliva, tears, okay, mucus at the bronchial surfaces, genitourinary tract, digestive tract. So at all these mucosal sites, you find IgA predominantly. Obviously, you can find IgG, but IgG, if you find at these sites, probably has to be because you might have some loss in integrity, and eventually, so basically because of the this antibody was present in blood, so through circulation actually it got there. Okay, but for IgA, so you don't require any such rupture in the integrity of membranes, and okay, so it actually has a way of getting to external secretions. IgA. Okay. Now you could actually consider that if you have to protect yourself against a mucosal infection, okay, the most common antibody isotype you would want to induce is IgA. Okay. For example, you have activated your B cells in a way that now it start producing these IgA type of antibodies. Those IgA actually can be found at most of these mucosal surfaces, and they can actually neutralize the presence of this virus, bacteria, parasite at this portal of entry. Okay, this is a very critical antibody for protecting you against an infectious agent, because of the fact that this actually is present in these secretions. Okay, structure of IgA. So you already know that you have this existing as a dimeric form, which is actually brought about by this J chain. Then you also have something that we call as a secretory component. Okay, which actually is again having this immunoglobulin domain-like structure. So it has four domains, 5 domains, 1, 2, 3, 4, 5. Okay. Now, there is a reason why secretory component actually is present in IgA, and that we'll talk about in the next slide. Okay. So this exists in dimer and sometime in tetrameric version. You find a lot of it in secretions, because it actually has a way of getting into secretions, and that way actually is that mechanism is brought about by having this secretory component. Okay, this purple color chain that you have. Okay, so this is where it is coming from, the secretory component. Okay, so let's say you have this epithelial layer on this side. So let's say you have this gut lumen. On the other side, you have the submucosa, where you have these B cells and activation of these B cells in pious patches, and basically underneath these M cells and so on and so forth. All these things are happening here. So you actually you activated your plasma cells. These plasma cells were making this antibody, right? Now, this antibody, which are produced there at these mucosal sites, they actually can interact with a receptor. This receptor we call it poly-Ig receptor, polyimmunoglobulin receptor. This is present on these epithelial cells, right? And once this receptor interacts with the constant region of IgA, so this basically followed by this interaction, it is internalized in these vesicles. And because of the presence of these enzymes in these vesicles, so some part of, so basically this is cleaved here, some part of it remains here, and some part of it actually get associated with this antibody molecule, right? And this facilitates basically its transport across this epithelial cell membrane layer. Okay, from submucosal side to the lumen side, this antibody can migrate by binding to this poly-Ig receptor. This process is called transcytosis. Okay, same process is actually utilized when you find this antibody, this IgA antibody in breast milk. Okay, so and now you know the origin of this secretory component. So this actually is coming from this poly-IgR, which is present on these epithelial cells. A portion of it actually is nothing but secretory component. Yes. All right. So IgE. it's very powerful antibody in its biological functions. Okay, usually exists in very low concentrations. Okay, um, as I said, it's very critically involved in type 1 hypersensitivity reactions or allergic responses, right? Doesn't have this hinge region. So instead of hinge region, so you have this additional domain. Okay. Um, for all these allergic responses like hay fever, asthma, hives, anaphylactic shock, IgE antibody actually is the one which causes all these functions or these diseases, right? How? This is how it does it. Okay, let's say you have these mast cells. Okay, mast cells have these preformed granules. These granules contain histamines. Lot of histamine is present in these granules in mast cells, right? They are already there. It's just that you require a trigger, and they can be these histamine molecules can be released very efficiently out of this cell to cause allergic responses. How does it happen? So, for example, if you are able to induce this IgE kind of antibody, right? Your B cells are producing this antibody. So it utilizes this Fc portion, Fc epsilon, and receptor for this Fc epsilon are present on mast cells predominantly. A lot of these receptors are present on mast cells. Fc receptor specific for IgE are present on mast cells. Once these antibodies are there, doesn't matter whether the antigen is present or allergen is present or not present, so they can actually come and bind to these receptors. Okay. Usually they remain empty because the antigen is not hanging around. Antigen is not present, so they are there, but they actually are present. They are bound on the surface of these cells. As soon as you encounter this antigen, or in other words, we call it as allergen, for example, dust, mites, pollen grains, all those things, right? So they actually cross-link these two antibody molecules bound through Fc receptors. And these Fc receptors are specifically there for IgE. Once you have this cross-ligation of the antibody molecules, because of the presence of this allergen, okay, so you induce this downstream signaling events, a lot of calcium signaling actually is taking place in these cells. And these granules, which were already having this preformed histamine, right? So this is released, and now you are actually in big trouble, because then you start showing these symptoms almost immediately. As soon as you get exposure to these allergens, these histamines are released, and histamines basically act on your blood vessels, right? For example, you get swelling in your respiratory tract, you have difficulty in breathing, so these blood vessels basically get um, uh, dilated, right? Rather constricted, right? And as a result of it, so you get all the symptoms associated with allergic responses. This is the mechanism. This is the underlying mechanism. And the only way you can take care of this acute situation is you neutralize those histamines, antihistaminic. You have to actually take these anti-allergic drugs, which are nothing but antihistaminics. Okay, so they can bind to these, they release histamine, and then you can have a restoration of homeostasis, and you can alleviate your symptoms that you have developed as a result of this allergic response. Uh, Tell me, okay, so we still have quite some time. No issues.
So,
Yeah.
so can IgE also take part in parasitic allergy, helping the eosinophils?
Well, yeah, obviously. Eosinophils actually also have a lot of these Fc receptors which actually can bind to IgE. Yes.
Fine, sir.
Yeah. But for eosinophilic induction, so you require parasitic infection.
And so the mechanism would be quite same,
More or
right?
less same, yes.
Fine, sir.
Okay, IgD, very limited. So very limited level of this antibody you find in serum. Together with IgM, it forms this receptor, okay, the membrane-bound IgG on mature B cells, okay. So basically on surface of your B cell, so you have IgM and you have IgD, okay. These are actually a part of the receptor. Now, the identity, the specificity of IgD and IgM, if they are present on one particular cells, will remain same. Okay. And if you have this one B cell which you have activated by providing this antigen, okay, and eventually then this is going to get activated, start proliferating, start making antibodies. So whatever the isotype of antibodies are produced by this activated B cells, the antigen specificity remains same. So you can have subtle changes that some isotype will bind to this antigen more efficiently as compared to the other, right? But the antigen specificity remains same. Although, yeah, so two more things that you have to know is that this actually is required for a B cell activation process, because this is actually present on the surface and serving as the receptor, right? No particular biological function has been ascribed to IgD, even until today. I don't know, so if you can pull out some very recent literature to show what does it do, but not well-established function is associated with IgD. For example, for IgG, we know that if something need to be neutralized in serum, in blood, so IgG is the one. If something need to be neutralized at mucosal sites in secretions, so you have to have IgA. For inflammatory reactions, so you have IgE. IgM, it can be found in secretions, and also predominantly in IgM, the first antibody isotype that you form, because it can actually bind to multiple antigens present on this antigen. So this is also very good in agglutination and can efficiently activate complement system, right? Affinity of interaction will always be poor for IgM than for the class switched antibody that you form. It's either IgE or IgG, right? And this is because the initial isotype is IgM, and then there are some changes that have to happen also in the variable region to increase the affinity. This is called affinity maturation. We'll talk about that. Okay, but broadly we have talked about different classes of immunoglobulin that you find either in secretions or in circulation or at mucosal sites or elsewhere, right? Yeah, so yeah, this is actually a long table, and I'm pretty sure this is pretty boring as well. So I would suggest you go through this table when you have some time available. So basically it summarizes all these parameters for different antibody isotypes or different classes of antibodies, okay? So the molecular weight that you have, the heavy chain which is utilized by these different antibodies, serum concentration, what is the half-life of each one of those antibodies, and that will actually tell you which antibody response actually is more durable as compared to the other. For example, people keep on talking about that because of this coronavirus vaccination, so you had protection very early on, but now these vaccines are not protecting. And as a correlate of protection, what they are measuring actually is the level of these antibodies. And we know from this table, right, that doesn't matter what. After some time, the level of this antibody has to go down, right? IgG is the one which actually persists in your for longer than any other isotype. But this also maybe within 3 weeks time, so it will go down almost 50%. Okay, and if you wait for another a month, so it's almost 1/4. So you know that antibodies actually have to go down. So you cannot really keep flooding your blood with these antibodies, right? So, and that actually is the thing that we talked about. The purpose of vaccination is not to just make antibodies, also to make immunological memory. The memory cells which can generate antibody whenever you get the real exposure with that infectious agent. That's the purpose. Because we know with antibodies, so they have this half-life. After some time, they are going to go down. Doesn't matter what. Okay. Also, other properties are basically unlisted here. So whether they are able to activate complement system and to what extent, right? Whether they can cross placenta present on the surface of membrane. So in this case, you have IgM and IgT, right? Which can bind to Fc receptors responsible for phagocytosis. So IgG. Okay. Mucosal transport, IgA. Okay. Degranulation of mast cells, IgE. So these are some of the properties which actually are, this table actually is crucial in the sense that it talks about everything about antibodies, almost everything. Okay, go through this table at your available time when you have it. Now, the other thing that we need to talk about in this lecture actually is the antigenic determinant on immunoglobulins. Okay, antigenic determinant basically is the epitopes present on antibody molecule, which can be recognized by B cells, and then you start making antibodies against your antibody molecule, right? So there are 3 types of these antigenic determinants. One of them is isotype, we already know, right? Then you have the allotypes, and then you have the idiotypes. Rather, there is one very popular theory in immunology, idiotype, anti-idiotype theory, which actually is was proposed to regulate immune networks. Looks like somebody else is logging in, so we have to actually stop. How can it be? It's only actually 44. I think we cannot really stop it now, right? If we want, we can continue on it.
Yes, sir.
Anybody can start logging in at any time. So we almost have 16 minutes left for this lecture. So for 5 minutes, we'll try to complete it. Okay, isotypes, allotypes, idiotypes. And these antigenic determinant, we call them as isotopes, allotopes, idiotopes. Okay. And this will be a little more clear here. For example, isotypic determinants, they are actually present in the constant region of heavy and light chain. Okay, so these are some of the epitopes that, for example, you have, right? If you compare, let's say, IgG molecule and IgM molecule, so these are actually unique epitopes which are present in the constant region. Okay, let's say if this is the kappa chain, this is also the kappa chain, so you have the same antigenic determinant here. Okay, but gamma 1 and mu are different chains. So that means the epitopes which will be contributed by usage of amino acid in this region are going to be different. Okay. just read this statement, that constant region determinants that collectively define heavy chain, class and subclass, and each light chain type and subtypes within a species are called isotypic determinants, because they are also present in the light chain. Okay, so you got the point, that these are the antigenic determinant which are present in the constant region of an antibody molecule. Now, depending on what heavy chain you're utilizing, so these are going to be different. This is called isotypic determinant, right? In other words, so you call them as isotopes. It's not like a radioactive isotope. Okay, so it's like isotope, the antigenic, the other name of isotypic determinants. Allotypic determinants, basically within the same of, for example, within the same species. Okay, these are also present in constant region, occur in some but not in all members of the same species. Okay, so for example, you have mouse. This is a mouse species, right? Mouse IgG1, mouse IgG1. You have one strain, you have another strain. Some will be shared. Some of these epitopes will be shared. You have the same epitope here, same epitope here, same epitope here, same epitope here. But some of these epitopes, depending on what strain of animal you are using, so are different. So for example, this has a unique epitope, this has a different type of antibody, a different epitope, right? So against this, you will form an antibody, and against that, you will form an antibody. So if you form an antibody against this will only react to this particular IgG and not to this. So now if you categorize based on the cross reactions, so isotypic determinants are more immunoreactive than allotypic. And finally, you have the idiotypic determinant, which actually are present in the variable region, which is basically shown here. So they are actually present in the variable region. Okay. So for example, although this portion is also called as what? What do you call this portion which bind to the antigen? Paratope, right? Paratope can also serve actually as idiotope. Okay, so the paratope that you have can also, the paratope and idiotope actually can be same. This is what I mean to say. And for example, if this antibody you inject in mouse, or for example in human, or even within the same strain, okay, so you actually can still induce some level of immune reactions against the antibody. So for example, if I isolate antibody from Vishnu and inject in another student, right? So you are going to the recipient will have antibody which are generated against the antibody molecule. Those will be called as allotypic determinants. Idiotypic are within the same individual. Okay, so for example, I have one antibody, and because of the formation of these antibodies, some epitopes were uniquely now constituted, and they actually can be recognized by my own B cells, right? And then you can keep on doing it, and this network was proposed to actually control immune regulation. This is called Neil Yarn, J-E-R. Some people call it as Yarn. Okay, but this is Yarn. Neil Yarn, so he proposed this theory of anti-idiotypic, idiotypic anti-idiotypic networks for antibody formation and immune regulation. Okay, I think now we can actually stop here. So we'll continue talking about BCR and the immunoglobulin superfamily molecules and so on and so forth. All right, so we'll continue from here in tomorrow's class.